Thanks to the organisers for this opportunity to talk at what's already been a really exciting meeting. So I want to start by talking about membrane tension and a pathway that we've identified through which low membrane tension can actually regulate contractility and retraction in cells that are migrating rapidly. And then I want to kind of move on later on to some newer ideas where we think similar pathways can actually contribute to matrix assembly in the context of metastatic niche assembly. So that's this supporting matrix that surrounds tumour cells within metastases. So membrane tension is a force that resists deformation. And the apparent membrane tension that cells feel is really made up of the in-plane membrane tension together with connections between the membrane and the underlying cytoskeleton. So just like pushing on a balloon, when you push on a membrane, you increase membrane tension. And for a long time, it's been thought that membrane tension, just like in this balloon example, would propagate really rapidly around the structure. But this view has been challenged in recent years. First of all, data from Adam Cohen's lab at Harvard, and what they did was to establish two membrane tethers from the same cell, pull on one tether and show that you didn't see an increasing membrane tension in a second tether. And what we've found is that membrane tension can be low specifically at the rear of fast-moving cells, particularly in 3D. So membrane tension can be different within different regions of the same cell. So we got to this by analysing the way that cells, cancer cells, move in a 3D matrix. And we use cell-derived matrix type systems like this. This is a thin layer of fibrillar collagen and fibronectin that cells move in and along the fibrils, but we also do this in thicker plugs of collagen and fibronectin rich extracellular matrix, which is very similar to that environment that we find around the metastatic tumours. Here you can see a cell moving in this 3D matrix. And what you'll notice is that this retraction phase of the migration cycle is really quite strikingly fast, especially when we compare it to the protrusion here. These cells, as they're moving in 3D matrix, establish cell matrix adhesion complexes towards the front of the cell and under the cell body, but not around the rear. And in fact, we see other markers like caveolae, which I'll come back to a bit later on. So as these cells are moving in 3D matrix, they're actually strain stiffening the matrix in front of them. So they're pulling on those matrix fibers, and in a way, they're kind of generating their own duratactic type of gradient to move towards. So we've also looked at membrane markers that cells are moving in 3D, and this is what brought us towards membrane tension. So in this movie in the top left, you can see membrane GFP, and it's really accumulating around the rear of this cell as it's moving in 3D. DIL is a dye that intercalates into the membrane, and here you can see at the rear of the cell, as this cell kind of squeezes forward, the membrane concertinas in at the back. So together with Eva Palucci's lab at the time in UCL, we wondered whether actually this accumulation of membrane could change some of the properties of the membrane at the rear of the cell. So we first used an optical trapping method in cells that were on rigidity gradients, so pulling membrane tethers from the rear or the front of the cell, and we found really strikingly that we always see lower membrane tension at the rear of cells moving on rigidity gradients. So we don't see this striking difference in membrane tension when we look at cells on uniform stiff gels, acrylamide gels, but we do see it when we look at cells in 3D matrix. So here you can see one cell that's moving up towards the top right and the second moving down towards the bottom left. We've used flipper TR, which intercalates into the membrane, and we can read out on membrane tension by measuring the fluorescence lifetime of this probe. And essentially we see low lifetimes and low membrane tension, specifically at the rear of these cells. These are cells on cell-derived matrix, this thin layer of fibrillar collagen and fibronectin. But we see the same things when we look deep within plugs of extracellular matrix. So here, cells are escaping from this spheroid over here, moving out into the matrix. And again, we can see low membrane tension as read out by the lifetime of flipper TR. So membrane tension's low at the rear of fast-moving cells in rigidity gradients and in 3D. So we got interested in these structures called caveolae because people had already shown that they're found at the rear of the cell. They can modulate or mediate the endocytosis of cargoes, but they'd also been shown to respond to membrane stress. So for example, in endothelial cells under shear stress, caveolae will kind of flatten out and release a reservoir of membrane to buffer membrane tension. So we wondered whether the reverse was true, and perhaps caveolae could form in response to low membrane tension, and that's why a lot of people had spotted them at the cell rear. So in our systems, we see caveolae at the rear of the cell. So here you can see a marker, cherry caveolin 1, specifically at the rear of the cell that's moving up towards the top right. And because it's membrane associated, we see it at the Golgi 2, but it's really only at the membrane around the rear of the cell. Other markers for caveolae, we can see decorating the rear of cells. So this is Cavin 1, and it's a maximum intensity projection. So these puncta are actually kind of coating the outside of the rear of the cell. And again, when we look in thicker plugs of extracellular matrix in spheroid type experiments, cells that are escaping from these spheroids show accumulation of Cavin 1, this caveolar marker. So caveolae can form around the rear of the cells, but has this got anything to do with membrane tension? So to begin with, to get at this, we decided to use osmotic shock, which essentially is not the most elegant method. We're adding water to the medium, but what that will do is swell the cells and increase membrane tension. And we're using 50% water in these experiments. So here you can see M. cherry caveolin 1 localizing to the rear of the cell. This is the portion that's associated with the Golgi. And what we'll do is to flow in osmotic shock medium and then follow this up with isotonic medium to see if we can recover it. So first of all, what you can see is that the caveolae disappear under osmotic shock, and the cell kind of stopped moving then as that osmotic shock medium was present. But when we flow in the isotonic medium, the caveolae reform really quickly, and the cell kind of springs off in this forward direction again. So it looks like osmotic shock can oppose caveolae formation at the rear of the cell and stop the cell moving in a really dynamic timescale. We've also used constitutively active esrin as another way to try and increase membrane tension. So constitutively active esrin will cross-link the membrane to the underlying cytoskeleton, and we see it localized really tightly around cells that are trying to move in 3D matrix. Constitutively active esrin will oppose the differential membrane tension that we see in cells on rigidity gradients. And again, constitutively active esrin prevents rear forward movement, and it prevents the localization of caveolin and cavin markers around the cell rear. So it seems like caveolae are indeed responding to low membrane tension and forming at the cell rear. But what we next wanted to get a handle on is how this might contribute to cell motility and rear retraction. So we used FRET probes to read out RhoA activity, and you can see, as you'd expect, we have high levels of RhoA activity, specifically around the rear of cells moving in 3D matrix. However, if we knock down caveolin 1, we can decrease the level of active RhoA at the rear. And similarly, if we use the osmotic medium trick, we can decrease RhoA activity, specifically around the cell rear. So it looks like low membrane tension and caveolae are required for RhoA activity at the cell rear. And to cut a little bit of a long story short, we identified ECT2 as a RhoA geth that's required to activate RhoA at the rear. So ECT2 is required for cell motility in 3D matrix, and it's required for the retraction, that rapid retraction phase of the migration cycle. ECT2 knockdown cells can't activate RhoA, so this makes it a really good candidate for perhaps a caveolae associated protein that could mediate rear retraction. So ECT2 is mainly associated with mitotic rounding and cytokinesis. ECT2 is actually found in the nucleus and cytoplasm of interphase cells. It accumulates around the rear of cells moving in 3D, and we see really nice co-localization with caveolae markers. So it looks like ECT2 can be recruited to caveolae. We see this enrichment of ECT2 around the rear of cells moving in 3D. But when we knock down caveoli, or caveolin 1 rather, to disassemble caveoli, we lose this recruitment of ECT2 to the cell rear. So we think that low membrane tension allows the formation of caveoli, which in turn recruit ECT2, which can activate RhoA to promote contractility and rear retraction. But that's quite a linear pathway that I've described there. We also did some mathematical modeling that led us to consider the idea or predict that positive feedback could be a feature of this cycle. So, so more contractility we might predict around the cell rear could lead to a further decrease in membrane tension and more caveoli formation, which could further increase contractility. So we decided to test this first of all using the ROCK inhibitor Y27632 to inhibit contractility. And so if this feedback loop is in place, what we'd expect is that inhibiting contractility would affect the localization of caveoli at the cell rear. So if you look at the movie on the right, that's probably the best illustration of this. As we flow in the ROCK inhibitor, the caveolin kind of melts away, internalizes, and the cell stops moving. So this is a global inhibition of contractility that redistributes caveoli. And what we wanted to try was something that's a little bit more focused. And in order to do this, we used a caged form of cytokinesin D. So what we've done is to uncage this right at the rear of the cell. So we release this to interfere with F-actin. And again, the movie is quite illustrative of this. So I'll uncage here at the beginning of the movie, and then you can see what happens to F-actin, which decreases in intensity over time. And we also lose caveoli at the rear. So this suggests if we perturb contractility or F-actin, we lose the localization of caveoli, really giving us a good indication that positive feedback is part of this system. Okay, so that was a quick kind of introduction to this idea that low membrane tension is a mechanical signal sensed at the cell rear by caveoli, and these form to recruit the RhoA GEF ECT2, which in turn activate RhoA and contractility. And the more contractility you generate, the lower membrane tension you can induce to further recruit caveoli and activate RhoA. So that's what we think leads to that snapping forward of the cell rear as they're moving in 3D. But now I want to kind of try and convince you with some preliminary data that this pathway might also play a role in matrix assembly. So we're interested in matrix assembly within the ovarian cancer metastatic niche. And ovarian cancer is now thought to arise in the fallopian tube, progress via the ovary, which is an important staging post, but the real problems stem from metastasis within the peritoneal cavity. And the omentum is a fold of peritoneum that seems to be particularly important in terms of the metastatic spread of ovarian cancer. So here are some metastatic cells within the omentum, and you can see these blue cancer cells are surrounded by a stromal matrix, which is rich in collagen and fibronectin. These matrix proteins I've already mentioned that we've been modeling cells migrating through. So what we're trying to try and understand is how this matrix is formed and how it can support cancer cell proliferation, survival, chemoresistance, and further metastasis. So we can kind of think of extracellular matrix molecules as building blocks. So fibrillar collagens, for example, are the major structural components of interstitial and connective tissues like that we find in the tumor stroma. But there are also other matrix-associated proteins, including glycoproteins like fibronectin, which I've already mentioned. But this whole kind of cocktail of stuff comes together to produce a really complex tumor microenvironment that recruits growth factors and matrix ligands, which provide signals to the cell. And it's these signals that elicit the proliferative, pro-survival, chemoresistance type signals characteristic of cancer, and in particular in ovarian cancer. So why do we think that any of the stuff that I might have talked about before is important here? Well, it turns out that matrix assembly requires actomyosin within stromal cells. Actomyosin contractility can actually change the conformation of matrix molecules and help to assemble a higher order matrix structure. So just to illustrate this, we've got a couple of points on this slide. So first, we've been really trying to build on work from Fran Borkel's lab, where they characterized a desmoplastic response in the high-grade serous ovarian cancer metastatic niche. And we can also see upregulation of matrix proteins in the disease burden dimension when we compare it to the uninvolved omentum next to the diseased part and omentum from non-cancer patients. So we see in particular upregulation of collagen 11s, fibronectin, and versican. And we're interested in these three proteins, but I'm going to focus on fibronectin for the rest of this talk, because fibronectin is known to be a scaffold for the formation of fibrillar collagen matrices. So mouse tissues that lack fibronectin are unable to assemble collagen networks. So what we've done is to isolate cancer-associated fibroblasts from metastases, and you can see the membrane of one of these CAFs labeled in magenta here, and we add labeled fibronectin to these cells, and we can watch them remodel the matrix. And what you'll see in this movie when it plays on the right is the extension of a matrix fibril just from one end. So hopefully what you can see here is the matrix fibril growing out from one end. It's not that the whole matrix fibril is sliding across, it's really growing, and it grows about 6 microns in 10 minutes. So this is a really fast and really dynamic process. And the timescales are very similar to what I was talking about in terms of retraction earlier on. So why might this be linked to contractility and membrane tension? Well, we know that when fibronectin, which circulates as a dimer and is secreted as a dimer, when it binds to integrins at the cell surface, those integrins can exert force on the fibronectin dimer because they're connected to contractile actomyosin. So as you pull on the fibronectin dimers, you open up the molecules, reveal cryptic binding sites, and that allows this fibrinogenesis process to occur. Because the integrins are transmembrane, we wondered if there could be alterations in the membrane as the integrins are pulling on fibronectin on the extracellular side, but being held by actomyosin on the intracellular side. And as the integrins are pushing and pulling through the membrane, perhaps you'd wrinkle and fold that membrane, and maybe you'd start to stabilize structures such as Caveolae, which I already mentioned can feed into the activation of RhoA through recruitment of GEFs. So maybe low membrane tension could feed into this actomyosin contractile network that we need to generate fibronectin fibrils. So we first of all used FLIPper-TR to try and understand if there are differences in membrane tension in CAFs that are assembling matrix. So here in the top left, you can see cells assembling a far red labeled fibronectin that we've given them on their surface. And you can see regions of fibronectin fibrinogenesis, such as this RhoA here, and other regions where there's no assembly of that matrix. If we then look at the lifetime of FLIPper-TR within these regions, what you can notice is the blue pixels, the low lifetimes, co-distribute with areas of matrix assembly, whereas where there's no fibronectin assembled, we see higher lifetimes. So that really suggests to us that membrane tension could be low where cells are generating new fibronectin fibrils. But what would this mean for what could this mean for matrix assembly in CAFs? So we decided first of all to use the hyperosmotic shock trick that I showed you earlier. So again, we're adding 50% water to the medium, but we're doing this first of all by allowing cells to assemble a red labeled fibronectin and then flowing in a green labeled fibronectin together with osmotic shock. And when we play the movies, what you'll see is a flash as the labeled fibronectin is added, and then first of all in isotonic medium, the cell will start to generate matrix fibrils, but then when osmotic medium and green labeled fibronectin is added to the same cells, you don't see the same level of fibronectin fibrinogenesis. So that suggests that hyperosmotic shock can oppose fibronectin assembly in CAFs. We've also used the constitutively active esrin that I mentioned earlier. So here you can see fibroblasts expressing constitutively active esrin are really having a hard time assembling fibronectin fibrils. And where they can start to assemble fibronectin, we don't see low membrane tension. And that's in contrast to the situation, this control down here, where cells are assembling fibronectin more readily, and we see blue pixels and low lifetimes indicating low membrane tension where fibronectins and fibrils are being formed. So these data really suggest to us that low membrane tension is required to allow fibronectin fibrillogenesis. And just the last little bit of data I wanted to share with you, again, quite preliminary, but what we can see is when we look at cells or CAFs assembling fibronectin in magenta here and stain them for caveolin 1, we can actually start to see caveolin 1 kind of lining up along these newly forming fibronectin fibrils. So that suggests that perhaps this pathway that can sense low membrane tension in retraction and migration could also be operating in CAFs as they assemble matrix. So just to summarize this second part, we think that matrix changes in the metastatic niche reflect an expansion of the stromal compartment through matrix assembly. Fibronectin is a good example of a scaffold for assembly of new matrix. We think that low membrane tension and caveolae coincide with areas of fibronectin fibrillogenesis, and they could actually lead to the same sort of contractile signaling we see at the cell rear, which would be required to generate those fibronectin fibrils that form initially on the surface of CAFs to allow assembly of a higher order matrix niche that ultimately forms the tumor microenvironment for high-grade serous ovarian cancer metastases. Okay, so we'll stop there. I'll just thank Matt, Joe, Eleanor and Soledad, who did most of the work that I've presented today. A whole bunch of fantastic collaborators in Manchester, in the UK and around Europe. And I'll stop there. Thank you.